Welcome to the deep dive. If you're prepping for an exam in anatomy, maybe physiology, or you're just diving into your first histology course, you know how overwhelming it can be.
Oh, yeah. The volume of material is just immense.
But there is a secret shortcut, and it's that everything in the human body, everything from the brain to your bones, it all boils down to just four basic building blocks.
That's the core realization, and that's exactly what we're focusing on today. Our source material is that foundational chapter from histology, a text and atlas, the one that deals with the concept and classification of tissues.
Right.
So our mission today is to walk you through every concept, every classification rule, and every visual cue in that chapter, step by step.
And we want to make the distinction between, epithelium, connective tissue, muscle, and nerve tissue.
Yeah.
Completely crystal clear.
Yeah, we're not just giving you a summary.
No, we are breaking it down as if you were looking at the diagrams and the micrographs with us. By the end of this, you should have a really solid grasp of the structure, the function, and even where these tissues come from.
The big question we're wrestling with is, how does the body achieve this incredible, mind-boggling complexity using only four fundamental blueprints? Understanding the rules of organization is really the key to mastering the whole subject.
Okay, let's unpack this and start right at the beginning with the foundational definitions. So when we define a tissue, we have to talk about organization. The textbook says tissues are aggregates or groups of cells organized to perform one or more specific functions.
And that word organized is so important. It tells you right away that tissues aren't just, random clumps of cells.
Right, they're not just a pile.
No, they are functional, collaborative units. And this kind of forces a shift in how you think about the body's hierarchy. We always call the cell the basic functional unit, and, that's true in a way. It performs all the processes of life. But it's really the tissues, through that collaborative effort, that maintain the functions of the entire body.
The single muscle cell can't lift a weight.
Exactly. But organized muscle tissue can. It's the collaboration that matters.
And for that collaboration to even be possible, the body needs some pretty specialized mechanisms.
It does. The textbook points out a few. You've got these specialized junctions that connect the cells, which are absolutely crucial for just holding things together.
Like a biological superglue.
It really is. And that's just the physical part. For them to work together, they have to communicate constantly.
Right.
So they use other specialized junctions, like gap junctions, to allow for a unified operation.
To a what, like passing signals back and forth?
Instantly. Think about the cells of the heart. They all have to contract at the exact same moment. That's only possible because gap junctions let electrical signals just fly between cells that are touching.
Okay, so beyond the physical connections, tissues also have to be responsive.
They need specific receptors on their membranes that are just waiting for a signal.
Like a hormone or a nerve impulse.
Or even just mechanical stress, like pressure. This lets the tissue act as one cohesive, coordinated unit.
And this coordinated effort, whether you're looking at a kidney tubule or the lining of a blood vessel, is all done by combining just four basic tissue types.
Just four. It's amazing when you think about it.
Let's run through those core four because, this is the basis for everything else we're going to talk about.
Okay, so first you have epithelium. It's your covering, lining, and secretory tissue. It forms surfaces, lines cavities, and makes up glands.
Okay, number one.
Second, connective tissue. This is the structural and functional bedrock. It basically underlies and supports the other three.
The framework.
Exactly. Third, muscle tissue, which is the engine of movement made of contractile cells. And fourth, nerve tissue, the body's information network. It receives, transmits, and integrates data.
Now, this is where it gets a little tricky, and the textbook highlights this really well, is what it calls the classification paradox.
Yes. This trips up so many students. We use two fundamentally different ways to define these four tissues.
It's not consistent.
Not at all. We define epithelium and connective tissue mostly by their morphology.
So how they look, their shape, their structure.
Right. But then you get to muscle and nerve tissue, and suddenly we're defining them by their function. Contractility for muscle, information transmission for nerve.
But the real twist, the irony here, is that when you go to subclassify them, it often flips completely.
It does. Take muscle tissue. We define it functionally, it contracts. But then how do we subclassify it?
Smooth muscle and striated muscle.
Which is a purely morphological distinction. It's just based on whether or not you can see those little stripes, those bands of proteins.
And the textbook gives this perfect example that just breaks the whole system. The myepithelium.
Ah, the ultimate curveball. This is a type of contractile tissue, so it absolutely functions like muscle. It helps squeeze secretions out of glands.
So it's a muscle.
Functionally, yes. But because of its location, it's right there with the epithelial cells of the gland. We classify it as an epithelium.
So that one example just proves that you can't rely on one simple rule. You have to understand both the structure and the function to get it right.
Exactly. Neither definition is absolute. You have to look at the context.
Okay, so let's dive deep into that first category, epithelium, the barrier layer.
The structure here is defined by two things you always have to look for: close cell apposition and the presence of a free surface.
Close apposition just means the cells are jammed right up against each other.
They're contiguous, literally touching. There's almost no space between them except for those specialized junctions we mentioned.
And that tight packing is what lets it form a barrier.
A highly selective barrier. It controls what gets to pass from, say, the outside world into your underlying tissues.
So where do we find this stuff? When we say free surface, what are we talking about?
Well, the most obvious one is the exterior of your body, your skin, the epidermis, but also the outer surface of your internal organs and the lining of pretty much every internal cavity.
The pleural cavity around the lungs, the pericardial around the heart.
Exactly. And very importantly, they form the continuous lining of your entire cardiovascular system, and they also form all of your glands and their ducts.
And when it comes to classifying them, the rules are purely about how they look.
Purely morphological. You look at two things: the shape of the cells and how many layers there are.
The shapes are pretty straightforward. You've got squamous, which are flat like pancakes.
Then cuboidal, where the height and width are about equal, like little cubes. And finally, columnar, where they're much taller than they are wide.
And then for layers, it's either simple, just one layer of cells, or stratified, meaning multiple layers stacked up.
Simple epithelia are usually all about absorption or secretion, where you need things to pass through easily. Stratified epithelia are built for protection.
Okay, let's try to visualize this. The book has figure 4.1, which shows a few different types.
Right. So let's describe what you'd see. In figure 4.1a, you're looking at simple cuboidal epithelium, maybe from a pancreatic duct. You'd see a single layer of cells that look like a ring of little blocks. The key thing is that their height and width are almost identical. You can clearly see the empty space in the middle, the lumen, and the basal surface of the cells sitting on the connective tissue below.
And if we contrast that with figure 4.1b, which shows simple columnar epithelium from the gallbladder.
The whole profile changes. Now the cells are tall and slender, like columns. Their nuclei are usually all lined up in a neat row near the bottom of the cells. Their height is the dead giveaway.
Now 4.1c is stratified squamous epithelium, like from the esophagus. This one looks way more complex.
It is. You immediately see multiple layers of cells, which tells you its job is protection. But here's the detail that often catches students.
What's that?
Only the cells on the very top layer, the ones right at the free surface, are actually squamous and flat.
So as you go deeper.
They get rounder, more cuboidal, and if you look all the way down at the basal layer, right where it meets the connective tissue, it looks like a dark, intense band.
And why is it so dark?
That's a crucial visual cue. It means those cells are small and packed with a nucleus that takes up most of the cell. It's called a high nucleus to cytoplasmic ratio. These are the active stem cells, constantly dividing to replace the layers above them.
Okay, so beyond shape and layers, some epithelia have special modifications on their surface, right?
Absolutely. Tailored for their specific job. Simple epithelia might have tiny little folds called microvilli for absorption.
Like in the intestines.
Exactly. Or if they need to move stuff across the surface, like mucus in your respiratory tract, they have longer whip-like cilia.
And what about the stratified ones?
There, the big distinction is keratinized versus non-keratinized. Keratinized is what you have on your skin. It's tough, waterproof, and has a layer of dead cells on top.
And non-keratinized is on the inside, like the esophagus.
Right. It's kept moist, so it doesn't need that dead protective layer of keratin.
Okay. And finally, there are two absolute non-negotiable rules for all epithelia.
Two rules you can always count on. First, every single epithelium rests on a basal lamina. It's a thin, non-cellular layer that acts like double-sided tape, sticking the epithelium to the connective tissue below.
And the second rule?
They are all avascular. They have no blood vessels of their own.
Which is a huge functional problem.
It raises a big question, all right. If there's no blood supply, how do these cells survive? Especially the ones on the top layer of a stratified epithelium.
How do they?
The textbook explains it has to happen through diffusion. all the oxygen, all the nutrients have to soak up from the blood vessels in the connective tissue right underneath that basal lamina.
So that means epithelium and connective tissue are always found together. They're a mandatory partnership.
Always. And it also means there's a physical limit to how thick an epithelium can get. It can't get thicker than the distance nutrients can effectively diffuse.
Okay, so if epithelium is all about cells being packed together, connective tissue is the absolute opposite.
A complete 180-degree turn. Its defining characteristic is its extracellular matrix, or ECM.
So it's not about the cells, it's about the stuff between the cells.
Exactly. In connective tissue, the cells are conspicuously separated. They're not touching. And all that space between them is filled with this complex material, the ECM, that the cells themselves made.
So when we classify it, we have to look at both the cells and what that matrix is made of.
Right. And it all traces back to one embryonic origin, the mesoderm, that middle germ layer.
So when we look at proper connective tissue, the book divides it into loose and dense.
And loose connective tissue, or LCT, is the most common kind. You find it right under most epithelia. If we describe figure 4.2a, what you'd see is an ECM, where the fibers, mostly collagen, are all loosely arranged, kind of haphazard.
And lots of cells, right?
Very cellular. You have the main matrix-making cells, the fibroblasts, but you also have a whole bunch of migrant cells from the immune system wandering through. Macrophages, plasma cells.
So you'd see a lot of different-looking nuclei scattered around.
You would. Now, contrast that with dense connective tissue, or DCT, which is built for strength.
Wow.
Looking at figure 4.2b, the difference is night and day.
How so?
The collagen fibers are way more numerous, and they're packed together so tightly there's hardly any space left.
And fewer cells.
Much sparser. It's mostly just the fibroblasts needed to maintain those huge collagen bundles, so you see far fewer nuclei. And because of all that protein, it stains really intensely, a deep pink. It's also much less vascular than LCT. Its job is mechanical, not metabolic.
So beyond loose and dense, we have specialized connective tissues.
Right, and these are defined almost entirely by what's in their unique matrix.
Like bone.
The matrix in bone is mineralized with calcium and phosphate, which makes it incredibly rigid.
And cartilage.
Cartilage matrix is packed with water that's bound up in these special molecules, creating a firm but flexible gel. It's strong but resilient.
And then there's the weirdest one, blood.
The most fluid connective tissue of all. Its cells are just suspended in a fluid ECM called plasma. That fluid matrix is what allows it to circulate and transport things all over the body.
Okay, which brings us to another one of those fascinating exceptions. A classic exam question. Adipose tissue.
Ah, yes. Fat tissue. It's categorized as connective tissue, but it breaks the rules again.
How?
Because its defining features are all about the cells, the adipocytes, not the matrix. The cells completely dominate the structure. There's very little ECM.
And its function is way more than just storage.
Oh, absolutely. Adipose tissue is a vital endocrine organ. The adipocytes don't just sit there holding fat. They're actively secreting hormones that regulate your entire body's energy balance. So even though it's technically a connective tissue, its behavior is totally different.
All right, switching gears completely. Let's move to the functional tissues, starting with muscle.
With muscle tissue, we're defining it by one core property, the ability of its cells to contract.
And that contraction is driven by two specific proteins, right?
Two core proteins, actin, which forms the thin myofilaments, and myosin, which forms the thick ones. Their interaction, them pulling against each other, is what generates force.
And for that force to actually do anything useful, the cells have to be highly organized.
They're elongated, we call them fibers, and they're grouped into these distinct bundles. Crucially, their long axes are all lined up in the same direction. This ensures that when they all contract, the force is unified.
So we use how they look to subclassify them into three types. Let's use figure 4.3 from the textbook to visualize this.
Okay. Figure 4.3a shows skeletal muscle. When you look at these cells, they're huge, long cylinders, and two things should just jump out at you.
Okay, what are they?
First, the obvious repetitive cross-striations. These are the visible bands created by that super-organized arrangement of actin and myosin.
The second thing.
The nuclei. You see lots of them, and they're all pushed to the very edge of the cell, right up against the membrane.
Okay, now let's look at 4.3b, cardiac muscle.
It also has striations, like skeletal muscle, but the cells are different. They're individual, smaller, often branched cells, and they're arranged end to end.
And the nuclei are in the middle.
Usually just one or two right in the center. But the absolute defining feature, which you can see marked with arrows in the image, is the intercalated disc.
What is that?
It's a dark, thick line that marks the boundary where two cardiac cells join. It's a specialized junction for both adhesion and that super fast communication we talked about.
And finally, figure 4.3c shows smooth muscle.
Right, from the wall of the intestine. These cells are spindle-shaped with long nuclei in the middle, and the key visual feature here is what's missing.
No striations.
Exactly. The cytoplasm is smooth, the actin and myosin are still there, but they aren't arranged in that same highly ordered way so you don't see the stripes.
So it's worth pointing out something here. You said all cells have actin and myosin.
They do, for basic things like cell division or movement. But only in muscle cells are they present in such enormous quantities and organized into these incredibly precise arrays we call myofilaments. It's the sheer scale and organization that turns a basic cellular tool into the engine for macroscopic movement.
All right, our fourth and final tissue type, nerve tissue, the body's information processing system.
It's functionally defined by its ability to receive, transmit, and integrate information from, well, everywhere, inside and outside the body, to control everything.
And the main players are the nerve cells, the neurons, and their support system.
The neurons are the electrical specialists. They're built to generate and transmit electrical impulses, often over huge distances.
And their shape is all about that job.
Yeah.
We have the cell body with the nucleus and then these processes sticking out.
Right, you have a single axon, which is the long process that carries impulses away from the cell body, and they can be incredibly long.
Over a meter sometime.
Easily. And then you have the dendrites, which are usually multiple, shorter, branching processes that receive information and carry it toward the cell body.
The book makes a practical point, though. Under a standard microscope, it can be really hard to tell an axon from a dendrite.
Very difficult. You often need special stains to see them clearly.
So the critical point of communication happens at the synapse.
That's the specialized junction where the electrical impulse gets transferred to the next cell. And usually the transfer isn't direct electricity, it's chemical.
So it releases neurotransmitters.
Exactly. The electrical signal hits the end of the axon, which triggers the release of these chemical messengers, or neuromediators. They cross the tiny gap, bind to the next neuron, and that generates a new electrical impulse.
And that chemical step allows for modulation and control.
Precisely. Now, neurons can't do any of this alone. They're completely dependent on their supporting cells.
And those are different depending on where you are in the body.
They are. In the central nervous system, the brain and spinal cord, the supporting cells are the neuroglial cells, or glia.
And out in the peripheral nervous system, all the other nerves.
There, the support comes from Schwann cells and satellite cells.
And these support cells are not just passive packing material. They're incredibly active.
Vital. They physically separate neurons. They produce the insulating myelin sheath that makes nerve impulses travel way faster. They clean up debris through phagocytosis.
And in the brain, they form the blood-brain barrier.
Right, which strictly controls what gets from the blood into the brain tissue.
Let's look at figure 4.4 to visualize this. Figure 4.4a shows a peripheral nerve.
Yeah, and you can see it taught in two ways. It's basically a bundle of these thread-like myelinated axons, all held together by connective tissue.
In the cross-section, the axons look like little red dots. And what's that clear space around them?
Ah, that's the classic sign of myelination. That clear halo is where the fatty myelin sheath used to be. It's lipid-rich, so it gets dissolved during the normal tissue preparation process, leaving behind that empty space.
Got it. And 4.4b shows a nerve ganglion.
Which is just a cluster of nerve cell bodies outside the CNS. The neurons are those big spherical cells, and you can see them surrounded by the tiny, dark nuclei of the satellite cells, their personal support crew.
And a final point on origins. All of this, the neurons and the support cells, comes from the neuroectoderm.
Oh, from neuroectoderm. Even a few cells in the nervous system that look kind of epithelial, like ependymal cells, still retain some of those absorptive and secretory functions, which is a neat little link back to their origin.
Okay, so now we have to step all the way back to the beginning, to the embryo, and talk about histogenesis, the origin of these tissues.
Understanding which of the three primary germ layers gives rise to which adult tissue is, it's the ultimate framework for understanding everything from normal development to disease.
And the whole process kicks off with gastrulation, which forms the trilaminar germ disc.
Right. This three-layered foundation, the ectoderm on the outside, mesoderm in the middle, and endoderm on the inside, is the blueprint for every single tissue and organ in the body.
Let's start with the ectoderm, the outer layer. It handles the outside world and the nervous system.
And we can divide it into two parts. You have the surface ectoderm, which, as the name implies, gives rise to our surface layers.
The epidermis of the skin plus hair, nails, sweat glands.
Also the epithelia of the cornea and lens in your eye, the enamel of your teeth, parts of the inner ear, and even the lining of your mouth and the very end of your anal canal.
And then the other part is the neuroectoderm.
And this is huge. The neural tube part of it forms the entire central nervous system, brain, spinal cord, and so on.
But then you have the neural crest, which is just wild.
It's amazing. These cells are highly migratory. They break off from the neural tube and travel all over the embryo, forming this incredibly diverse range of structures.
Like the entire peripheral nervous system.
The entire PNS ganglia, nerves, Schwann cells, but also the hormone-producing cells of the adrenal medulla, pigment cells in your skin, the cells that make dentin in your teeth. I mean, the list goes on and on.
Okay, moving to the middle layer, the mesoderm. This seems to be the structure and movement layer.
That's a great way to think of it. It is the source of all connective tissue without exception. Bone, cartilage, blood, fat, you name it. It also forms all muscle tissue, striated and smooth.
And the entire circulatory system comes from here too.
The heart, all blood vessels, lymphatic vessels, including their lining, the endothelium. And it doesn't stop there. It forms the spleen, the kidneys, the gonads, the adrenal cortex, and the mesothelium, that special lining of our big body cavities.
And finally, the innermost layer, the endoderm.
The endoderm basically forms the primitive gut tube, so its primary job is to create the epithelial linings of all the structures that grow out of that tube.
So the lining of the whole digestive tract.
Most of it, yeah. Plus the epithelium of the big digestive glands that branch off it, like the liver and pancreas. The lining of the urinary bladder and the respiratory system. And as figure 4.5 in the book shows, it also forms the key epithelial components of the thyroid, parathyroid, and thymus glands, which all start as outgrowths from the early pharynx.
So this incredibly detailed breakdown of histogenesis leads us perfectly into a really cool clinical correlation in the book, ovarian teratomas.
Yes, and this is such a brilliant illustration of what happens when those germ layers go haywire.
The whole thing comes down to pluripotential stem cells.
Exactly. These are embryonic stem cells that can differentiate into any cell type from any of the 3 germ layers. Most cancers, they arise from just one cell lineage.
But teratomas break that rule.
They completely defy it. They arise from these pluripotential cells, and so the tumor itself can contain a chaotic mix of tissues from all 3 germ layers, ectoderm, mesoderm, and endoderm.
So you find mature, differentiated tissues, but they're just jumbled up.
Completely unorganized. You might find fully formed teeth, patches of hair, bits of nervous tissue, even segments of bowel, all just haphazardly thrown together in one mass. It's non-functional chaos.
And these tumors almost always show up in the gonads.
Right, because that's where you find these pluripotential cells. And the prognosis is very different depending on location.
Ovarian teratomas are usually benign.
Usually benign. They're often called a dermoid cyst. They're quite common in younger women. But testicular teratomas are rare and usually malignant, composed of much less differentiated tissues.
The micrograph in the book, figure F4.1.1, just drives home how anarchic these things are. At low power, it's just a solid, unorganized mass.
But when you zoom in on the different insets, you can instantly spot tissues from all our categories, all mixed up.
Let's walk through them. What are we seeing?
Okay, so in inset A, you see perfect, simple columnar epithelium lining a little cyst. That's an endodermal or ectodermal derivative.
And right next door?
Inset B shows dense, regular connective tissue looking just like a tendon. That's pure mesoderm. Then in inset C, you've got hyaline cartilage and developing bone spicules, again, specialized mesoderm. And then shockingly, inset D shows actual brain tissue with glial cells, a classic neuroectodermal derivative.
So that's all 3 germ layers right there.
All 3. And it doesn't stop. Inset E has cardiac muscle fibers. You can even see the intercalated discs. And F shows skeletal muscle fibers.
Just everything.
Everything. And the key takeaway for a student is that even in this complete chaos, the basic rules of histology apply. You can still recognize these tissues based on the core characteristics we've been talking about.
So, having gone through all 4 types, the real goal when you're looking at a slide is just quick recognition.
Right. The first thing you should always do is try to identify the aggregates of cells as one of the 4 basic tissues.
And the textbook gives a really simple but effective checklist for this.
It's like a little diagnostic flowchart. First question. Are the cells at a surface? If yes, you're almost certainly looking at epithelium.
Okay, next question.
Are the cells in close contact with their neighbors, packed tightly together? That's epithelium. Or are they separated by a bunch of intervening material? That's connective tissue.
And the third question covers the functional types.
Exactly. Do they belong to a special group with properties like contractility and an organized fiber arrangement, that's muscle? Or do they have that unique structure for transmitting electrical impulses? That's nerve.
If you can answer those three questions, you can identify almost anything.
It's a really robust strategy, so let's just do a quick final recap of the core takeaways.
Okay. Tissues are functional aggregates, and every organ is just a combination of the four basic types.
Epithelium, defined by tight cell contact, always on a basal lamina, it's avascular, and classified by shape and layers.
Connective tissue, defined by its extracellular matrix. It's the support structure, includes everything from bone and cartilage to blood and fat.
Muscle tissue, defined by contractility, driven by actin and myosin. We identify it visually as striated, like skeletal and cardiac, or non-striated, which is smooth.
Nerve tissue, the integrator, made of neurons with their axons and dendrites and their essential support cells.
And finally, histogenesis. Every single one of these tissues traces back to one of the three germ layers: ectoderm for barriers and nerves, mesoderm for structure and movement, and endoderm for linings.
So if we connect all of this back to the bigger picture, let's think about the adrenal gland again. We said the adrenal cortex comes from the mesoderm, and it makes steroid hormones. But the adrenal medulla comes from the neuroectoderm.
Right, from the neural crust.
So how does the fact that these two functionally linked parts of one organ come from totally different embryonic origins? How does that influence their integrated control over something like the stress response? And what challenges could that dual origin create when things go wrong?
That's a fantastic question. That idea that two halves of a single functional unit can start life with completely different blueprints is a remarkable example of developmental convergence.
It shows how the body builds complexity through these integrated relationships, even across germ layers.
A fascinating topic, and it really highlights why knowing this stuff, knowing the histogenesis, is so vital to understanding clinical function.
We really appreciate you sharing this material for a true deep dive. We hope this detailed exploration has given you the clarity you need to master the concept and classification of the four basic tissues.